Thank you, Dr. Muir, for that kind introduction. It is my great pleasure and honor to deliver the 2021 AGS Surgery Day Lecture. I'm humbled to be named on this list of highly accomplished glaucoma surgeons that have preceded me. I want to thank the AGS for putting on such an excellent meeting during these difficult circumstances. This meeting is always one of the highlights of the year for me. When Dr. Boudin shared with me the invitation to deliver this lecture after last year's meeting, I was excited to share this experience with all of you. However, no one could have foreseen the nature and magnitude of the global pandemic that was preparing to unfold. Therefore, I will be the first to deliver this lecture virtually, and hopefully I'll be the last. Under the rubric of glaucoma, there's a lot to choose from. What I've decided to discuss is what those of us involved in clinical research do to help drive innovation, some of the challenges we face, and some humble suggestions for improvement.
These are my financial disclosures. Many of them are relevant, but we work a little bit with everyone, so I like to think I'm beholden to no one. So what we do in the research part of our practice is mainly conduct clinical trials. And I've really enjoyed participating in all these mixed trials that have gone on over the past 10 years or so. And because we do so many trials, we really have a good sense of what works and what doesn't work. So we're often asked by sponsors to consult with them, doing things such as reviewing their protocols, helping draft protocols, a variety of different subjects. The why of it, however, is not only because it's fun and interesting to participate in these trials, and it's a benefit to our patients to be able to offer tomorrow's technology today, but really because I think we can do better. So imagine if this was you, a 65-year-old with real glaucoma on a decent number of medications per day. I would want my life to resemble much more what's described in this scenario than what was available in the past. Even today, we can offer to correct our patients' presbyopia, we can combine MIGS procedures, we can use sustained release medication to achieve a much better quality of life than what was available just a few short years ago. So in seeking counsel about what to talk about in this lecture, there was a consistent theme, and that was, talk about what you're passionate about. So what I'm passionate about is really being ambitious in our ultimate goals. And we need more and better surgical options. And the traditionalist in me wants to have good data to instruct us on how and when to use them. And we need to develop them efficiently. These examples I'm listing here, from the inception of the company to getting through their pivotal trial, Ivantis, for example, was 10 years and $100 million. And so in their pivotal trial, they had roughly 550 patients. That comes up to somewhere in the neighborhood of $175,000 per patient. And so we discussed being more efficient in our processes, even reducing the required number of patients in a trial from 300 to 200 saves unbelievable amounts of money and certainly saves time and makes our process more efficient. So if tomorrow you developed a great new surgical intervention for glaucoma, my guess is you probably don't have $100 million. And secondly, I don't think that you or I want to have to wait 10 years for the next great surgical intervention to be available to us. Hence the need to improve our efficiency. And the need to develop these new products is because the more products that we have, the more we're able to individualize therapy for our patients. We all have a wide variety of patients. They all have very specific needs. And when they have a need, we want to be able to fill it. And ultimately, this leads to increased focus on the patient's quality of life with better visual acuity, less amount of drops required per day, and improved ocular comfort. So if our goal is to develop procedures for a specific population with good safety and efficacy data in the shortest possible time for the least cost and have them reimbursed, there are a lot of stakeholders. And each group has their own specific needs and specific responsibilities. We'll do a deeper dive into the sponsor's needs and responsibilities as the talk goes on, but for patients, they need to want to be study subjects and participate in our trials. The doctors also need to want to participate and need to have a trial that they can actually recruit patients. The FDA has a very important role. One of the most important is creating guidance for what a company needs to do in order to conduct a successful clinical trial and ultimately offer approval. ANSI, the American National Standards Institute, an organization that most of you have never heard of, and I'd never heard of it either a few years ago, which really codifies standards for everything, from what constitutes the proper composition and thickness of auto glass to how many threads are in a screw. They also codify the definition of what's mild and moderate glaucoma. What efficacy analysis do we need to use to prove success? All these things are codified by ANSI, a very important organization. And then payers determine what actually we get to use in terms of what's covered. And they're obviously not operating in a patient's best interest. So a quick shout out to CoolDev here, who in 2014 had the first FDA AGS workshop, in which he brought many of these stakeholders in the same room for the first time. The FDA, industry, investigators, and glaucoma thought leaders all together in the same room, looking at ways to move the ball forward more efficiently. So we've accomplished quite a bit over the past 10 years. We've conducted multiple mixed trials, and I'll just highlight a couple of them. The Glaukos iStent, approved in 2012, really launched an era of MIGS surgery. There were some issues with the trial design that did not allow us to determine exactly how well the original iStent worked due to no terminal washout, but we know it worked well and it was employed in thousands of patients very successfully. The CyPass trial was a landmark trial, and not just because it was a large, multi-center, randomized trial of a new device. They really set the standard for trial design and execution. And most importantly, they created clear guidance for those who come after them. So if you have a product and you want to do a clinical trial and you have similar patient population and you want to do it combined with a cataract surgery, you have a clear path as to how to do that. Their inclusion-exclusion criteria are excellent. They're often copied by others, and they're codified in ANSI. They were the first to employ a terminal washout, so we truly understand how well our MIGS devices work. They were the first to employ predetermined outcome measures, that familiar 20% IOP lowering from an unmedicated baseline, and they were very efficient in their recruitment, which is why they spent the least amount of money. So what have we learned? In this particular population, which is a very specific population, mild to moderate glaucoma patients, which importantly represents 80% of all glaucoma patients, we have a very good understanding of the effect of cataract surgery on intraocular pressure. Almost to the decimal point, cataract surgery lowered pressure equivalently in the 3 studies. This is not something that requires meaningfully further study. We also understand very well the effect of cataract surgery plus the MIGS device, and the Hydrus performs slightly better than the others in this regard. We have a very good sense of the safety of these devices. All of these studies had 5-year extensions, and some of those are still ongoing. But there are gaps. We regrettably still don't understand the effect of these MIGS devices as standalone devices, and payers have taken advantage of that label to deny coverage. So this is where we are today. Moving forward, we need approval for standalone devices, and of course we need coverage. We also need guidance for those coming behind who may want to do a study that doesn't involve cataract surgery and get their device approved. So how do we get there from here? I'm going to give you a crash course, really a Cliff Notes version of a crash course in how the regulatory world works. So the FDA has classes based upon risk benefit, class 1, class 2, class 3. In class 1, you'll recognize the Cahook dual blade being in that class. And class 1 devices get approved based on having good manufacturing practices. They don't necessarily require clinical data to be approved. Class 2, you'll find contact lenses and aqueous shunts in the same class. Class 2 gets approved based on something called a 510K. In class 3, you have IOLs and MIGS devices in that class. And usually those are approved in something called a PMA trial, or these large clinical trials you're used to seeing. So the curiosity is really around class 2. Why are contact lenses and aqueous shunts in the same class? And the reason for that is the patient population that the aqueous shunts are designed to be used in. So those devices were approved in patients with neovascular glaucoma and those with failed traditional incisional surgery on maximum medications. So those patients are felt to have fewer options, to be in a higher risk group, and therefore the risk benefit is considered to be better than for putting an IOL in an otherwise healthy eye. So what is a 510K? What does that mean? So a 510K is a pathway to approval where one has to show that you have a device that's substantially equivalent to another device that's already on the market, a predicate device. And once again, it's very important that you have the same patient population that was used for approval and the same type of device. And if you have that, you don't necessarily need clinical data. But the predicate device has to have been around for a while and have significant data. So the OMID ClearPath is an example of that. So the OMID ClearPath was approved via 510K process. It used a barbell as a predicate. It's made of the same material as a barbell, has the same tube diameter as a barbell, and because New World Medical has significant experience producing quality products, it was approved without a requirement for clinical data. The Zen was also approved via 510K process using an aqueous shunt as a predicate. It does not drain to a reservoir, and therefore the FDA required them to submit data. When data is required, there's by rule a minimum of 50 patients required, and they had close to 100 patients in their trial. A PMA trial, these are these large clinical trials that you're used to seeing. It's a much more rigorous process. Again, all the MIGS devices are class 3 devices. So the patient population is not restricted. You can use whatever patients you'd like. But what's noteworthy is anything approved via PMA process cannot be used as a predicate device for a future device. And lastly, a PMA supplement is another path that a person could use. And you use a supplement when you've already conducted a PMA trial, and perhaps you have a significant design change, and the iStent W would be an example of that. The iStent W with the wide flange versus the original iStent, which didn't have a wide flange, was approved based on this process and did not require additional data. However, if you chose to change your indication, say from mild to refractory, or you wanted to go for a standalone, then you could use this path, again, if you've already done a PMA trial, but you might require significant data. And as discussed earlier, this is a very important thing because for each patient, the cost is very significant. So as we mentioned, somewhere between $150,000 and $175,000 per patient, and significant time. So perhaps going from a 300 patient trial to a 200 patient trial could really make a lot of difference in terms of our efficiency. So there are standalone trials that are ongoing at the moment. We participated in a few of those. And so my 2 cents as far as opportunities are concerned is once we've established the safety of a device, we should be able to power a study based solely on efficacy, and that would perhaps help us not need quite as many subjects to get the product approved. And secondly, we really could use an intermediate path. Right now we have a mild to moderate path, and we have this refractory neovascular glaucoma failed incisional surgery path, but it certainly would be nice to have a path somewhere in between where we could recruit patients that more represent the type of patients we see in our practice. But the FDA is committed to making the process easier. Jeff Shuren is the FDA Director for Devices, and in a meeting last month, he committed to looking for innovative ways to streamline the approval process. So one of the trials that we participated in that just released their results was the iStent Infinite trial. The iStent Infinite is a wonderful device. It's a really nice improvement and a welcome addition to our armamentarium. So there's a wonderful injector that has unlimited shots versus the limited shots we currently have. There's 3 wide flange stents, and you can see in the image the 3 stents placed in the angle. The patient population was a tough one. You'll recognize the failed incisional surgery on max meds population with 72 subjects, which should ring some bells. It's a familiar number. Interestingly, I found this worked much better than I would have expected. And if you look at the mean baseline characteristics of the study population, they were on an average of 3.1 medications and had a history of 2 failed incisional surgeries. Yet, 76% of the patients achieved a 20% or greater reduction in their IOP, and 50% achieved a 30% reduction in their IOP. And in my view, if something works this well in a difficult population, it really should work quite well in a more mild and moderate population. So let's talk about a real company with a real product that's trying to navigate this path. iStar is a company based out of Belgium. I've been working with them for the last couple of years. They have a suprachoroidal implant that is different than the CyPass, and that's made out of a soft, flexible material that is meant to have antifibrotic properties, and it fits into the suprachoroidal space. So what does their decision-making process look like? I mean, the first thing they consider is a 510K an option that will require fewer patients. Well, what indication to seek? Should they go for mild to moderate? Should they go for a standalone? They need to write a protocol that answers the appropriate questions. With a suprachoroidal implant, you need to answer questions around refractive changes, ECD loss. Most importantly, they need to write a protocol that's recruitable. We've had protocols in the past that were so stringent that we were only able to recruit 2 patients in 18 months. If you have things such as very narrow visual field requirements, ECD requirements, it would be very, very difficult to recruit patients, and you really shoot yourself in the foot. You'll at some point need to approach the FDA. And if you're not following the path of a previously approved product, there can be a lot of guesswork in the process. It can be difficult at times to get an if you do this, then you will get this result answer from the FDA. And of course, after that, you need to find sites and hire people and raise money to move forward. But then the fun starts. You get to go on these exotic trips, wear funny scrubs, ride in dangerous helicopters. And I found that many of these places have very, very impressive facilities. And the purpose of these trips really is, besides your friends criticizing your surgical talent, is really to learn the procedure, teach others the procedure, assess the device in its current form, and try to really perfect the procedure. All products go through multiple iterations before they settle on the design that will be used in a clinical trial. On the left are the different designs that the CyPass look through before they settle on their final design. And Mini-Eject is no different. They look at different tip curvatures, tip designs, handpiece designs before settling on the iteration that we feel is appropriate for a U.S. clinical trial. The Mini-Eject does have published data that is somewhat difficult population, patients with starting pressure of 24.6 on almost 3 medications, and improved tip pressure of 15.1 on 1.3 medications. There was no washout in this study, but we hope to see a study in the U.S. soon so we can critically evaluate the type of data that we've come to expect. A product that I'm really excited about is the PreserFlow. It's gone by many names in the past, the InFocus, I may slip up and call it that. But unlike some, I like filtration surgery. It's as close to a cure as we have at the moment. I just don't want to see too many of them on my OR schedule on the same day. So the PreserFlow is a 8.5 millimeter aqueous shunt with a 70 micron lumen that is meant to shunt aqueous into the subconjunctival space. We recently completed a 2-year randomized clinical trial in the U.S., and it currently sits on the desk of the FDA awaiting approval. But the reason that this device exists, obviously, is to lower intraocular pressure. But the second reason it exists is to avoid hypotony. As we all know, trabs can fail, or filtering surgery really can fail in 2 ways. Either the pressure can be too low or too high, and this should take too low off the table. In addition, it should improve the patient experience with faster visual recovery, less astigmatism, better ultimate vision, and fewer postoperative visits. And that should also improve the surgeon's experience with a shorter operation, fewer post-op visits. And in my view, it doesn't need to beat trabeculectomy on IOP, it just needs to be close. So, as I mentioned, they recently completed a PMA trial. It was a large randomized trial, randomized 3-to-1 in favor of the PreserFlow. It was a tough population of patients who had intraocular pressure between 16 and 39 on maximum medications. They could not have had previous conjunctival surgery, and the primary outcome measure was the familiar 20% IOP reduction. As expected, the trabeculectomy group performed somewhat better than the PreserFlow group with regards to that outcome measure. There's no surprise there. Importantly, there's no washout. And in a study with no washout, I feel like this is not the ideal outcome measure. If you have a patient who had a pre-op pressure of 18 on 4 meds, had the procedure done, and afterwards the pressure was 15 on no meds, I would consider that an overwhelming success. With this criteria, that would be a failure. So when it comes to studies that don't have washouts, especially a filtration surgery, and trabeculectomy is a titratable surgery. We are targeting a specific intraocular pressure, not a percent reduction. And if we look at the highlighted part of the chart here that shows the percent of patients who achieved an IOP of 16 or less on no medication, the ultimate achieved IOP I think is a better measure, and the PreserFlow acquitted itself reasonably well in this regard. These are the results at 1 year, which shows an average pressure of 11.2 in the TRAB group, 14.2 in the PreserFlow group. Both reasonable results. Both had good medication reduction. And just note the slope of the TRAB group over time, as I'll refer to that again later in the talk. So one point about the protocol, it's hard to have a crystal ball when it comes to writing protocols, but one shortfall this one had was it really didn't allow for the optimum discernment of visual recovery. I had several patients in the trial that had a TRAB in one eye and a PreserFlow in the other. There's no question as to what was the better patient experience, but we didn't have the data to really show that. as well as I would have liked. And fortunately, this study did show, as expected, a benefit with regards to hypotony. For those who haven't seen a PreserFlow procedure, this is one of my early study cases. This is the original double-step knife. Tenon's management is identical to trabeculectomy. The PreserFlow is inserted into the tract, and then one checks for proper egress of AGS. In all clinical trials, there are lessons learned and the procedure evolves, and this often affects the results. In the CyPass trial, for example, we had an inelegant inserter, and I believe that affected visual acuity results as well as the pressure results. In the PreserFlow trial, initially we were using a dual plane incision. We often found there was kinking of the tube, and we switched to a single plane incision. We were using a 0.2 milligram per ml concentration of mitomycin pre-soaked on sponges for 2 minutes. Most of us feel that's much too short and much too low of a concentration of mitomycin to be successful. In all studies, the surgeries themselves are recorded, and those who watched the videos noted that Tenon's management was inconsistent. We've already talked about the trial design being a little bit better to show some of the patient experience benefits. And finally, when you have the world's best trab surgeons that you recruit for your trial, guess what? They're really good at trabs, so you have the best trab results ever in your control group. As I mentioned, I'm somewhat passionate about filtration surgery, so let's discuss some pearls that can be used not only with the PreserFlow, but with traditional trabeculectomy. So number one, I believe you should set a very high standard for yourself. For me, I try to fix the pressure for good. And what fix to me means is a good pressure for life, no meds. That's the goal. And Tenon's management is really the most important aspect of that. I really try to make sure that the conj never beats me. So in that light, I believe that most people underutilize mitomycin. I always use 0.4 mg per ml for somewhere between 3 and 5 minutes, depending on the patient. One other thing that gets my attention is when people talk about different kinds of blebs or different procedures. A good trabeculectomy bleb and a PreserFlow bleb should look the same. It should be low, diffuse, and barely discernible. Trabeculectomies are meant to be titratable, so construct your scleral flap so it truly is titratable. It's rare that a patient needs a pressure of 10. Usually a pressure of 14 will suffice and have you avoid hypotony. As I referenced in the PreserFlow study, the trabeculectomy surgeon started with a pressure around 20 and slowly brought it down under control of the surgeon. And so filtering surgery can be a bit arduous, but they'll rarely fail with appropriate follow-up and management. When doing filtration surgery, it is important to do a very posterior dissection. So you'd have your scissors all the way to the septum. Here you see me inserting the corneal shields. In many ways, these are placeholders for me, but they do soak up some of the mitomycin. As you can see, I inject the mitomycin posteriorly and broadly, and I like to think to good effect. I open in 2 layers and close in 1. If you want a low posterior bleb, you have to bring Tenon's to the limbus. There are many ways to close it, but for me, 909 on a TG 160 needle is preferred. Good blebs don't have microcysts. They should be low and diffuse and barely discernible. So in conclusion, let's be ambitious. We have limited time in this lecture, but there are some incredible innovations in the pipeline, and I truly believe we're on the precipice of great things. Let's have high expectations, not only for what we can do in the future, but let's have high expectations for ourselves for what we can do for our patients today. We need to continue to innovate, but we need to do so more efficiently. The FDA is full of incredible people, and I believe wants to be a full partner in this process. So we need proactive collaboration. but it's incumbent upon us to better incorporate payers in the process. Finally, more research sites, by definition, improves our efficiency. So if you've ever considered getting involved in clinical research, by all means, get on board. Once again, I want to thank the AGS for asking me to give the Surgery Day lecture, and also thank all the great people who've supported me throughout my journey. Starting with my parents, even though the bid to be a professional tennis player didn't work out, had it worked out, I wouldn't be here today. I want to thank the great ophthalmology faculty at Hopkins for being so inspirational, that 15% of our class chose ophthalmology as a career. I want to thank the glaucoma faculty at WashU, starting with Bernie Becker, Mike Cass, Al Coker, Carla Siegfried, and my good friend Marty Wax, all who inspired me to go into glaucoma. George Alvarado, a great person, particularly if he liked you, incredible mind who set me up for success in my career. All the friends in the glaucoma community that supported me. I've always loved being an AGS member. I want to thank the staff and ophthalmology associates, great research coordinator, partners that support our work. We couldn't do any of this without you all. I've been blessed with a wonderful family. My wife Julie, who's also a physician, my daughter Hayden, daughter Chandler, son Grant, and the Chandler Grant thing is purely a coincidence. And I want to thank you all for your attention. Hopefully you learned something new and were in some way enriched and inspired. Thanks again.